Hello everyone, I'm Tong from the University of Hong Kong. Today, I'm glad to present our work on 3D annotation in transnasal endoscopy via depth estimation. Let us look at existing 2D surgical annotations first. During a minimally invasive surgery, usually an endoscope is involved in acquiring visual information from the patient's body. While displaying this content on the screen, surgeons may perform annotations by marking or drawing. Apart from facilitating communications, annotations can serve as anatomic bookmarks that reduce surgeons' cognitive burden due to improved spatial orientation and improved endoscopic navigation. As a result, it may potentially lower the learning curve and enhance surgical training outcome. However, existing approaches mostly give 2D annotations that are in 2D endoscopic view, which means they do not anchor to the patient anatomy upon camera movement. And this motivates us to think, can we actually make virtual annotations on 3D anatomy even when the camera moves in 3D? And the answer is yes, we can. Let's take a look at augmented reality in endoscopy. AR in endoscopy means the overlay of computer-generated graphics onto the endoscopic view. Therefore, we can in fact make annotations in a virtual sense and then merge the virtual world and the real world in a three-dimensional context. In order to put a 3D annotation at a correct world precision, it is essential to obtain accurate depth information in front of the endoscope. In usual surgical AR, preoperatively segmented and reconstructed 3D anatomic models are registered to the surgical scene, which may then be used as reference surfaces to read depth information. However, we need to bear in mind that 3D anatomic model segmentation highly depend on scanning quality and human segmentation skills, and the fact that soft mucosal linings are not clearly observable in CT scans. As a result, disparity between the surgical site and pre-op models may exist, and depth deduced purely from pre-op model may then be unreliable. Then, how can we get more reliable depth? Well, endoscopes are cameras. How about directly derived depth from vision? Talking about depth measurement, there are in fact well-established traditional vision-based or hardware-based solutions in obtaining depth. For example, depth can be obtained without much difficulty by stereo vision or active depth sensors. However, while our target surgical site in this study is nasal cavity, size constraint does not allow us to adopt these solutions. Visual SLAM is also a possible candidate to obtain depth. There is also a previous study that employed the famous monocular OP-SLAM, a feature-based visual SLAM, to perform tracking and mapping in gastroscopy. However, due to repetition of texture or lack of distinctive features in the nasal cavity, there is a high chance of getting erroneous results, or tracking may even fail to be initiated. Recent advancement of supervised monocular depth estimation by using convolutional neural networks has drawn our attention. These networks are trained to predict a depth map from an RGB input frame, with dense depth value estimated for every pixel. Though, we need to note the fact that a large dataset with ground truth depth labels is required for supervised learning, and it is usually impractical to collect ground truth depth using active sensors at the real target anatomy. Trying to address this impracticality of getting ground truth depth, we propose real-to-virtual domain transfer-based depth estimation in transnasal endoscopy. In this workflow, we employed a fully supervised monocular depth estimation network to predict frame-wise depth maps. What's different here is that the network is trained with RGB images and their corresponding depth maps that are prepared by 3D rendering in a simulator. In this manner, there can be an unlimited amount of ground truth depth available for training. Though bear in mind that during network testing, real endoscopic RGB images are fed into the depth estimation network instead of 3D rendered images, and they differ quite a lot in terms of image style. To address this issue, we employed an adversarial network that performed an image-to-image style transfer, such that endoscopic images are transformed into synthetic-like images prior to depth prediction by the monocular depth estimation network. For depth estimation training, we firstly reconstruct an anatomically accurate 3D nasal airway model from CT slices of a cadaveric head, and then we import it into a virtual world simulator. Then, we set up a virtual camera fed with physical camera intrinsic parameters of a real monocular Olympus laryngoscope. Both synthetic endoscopic images and their corresponding depth maps are collected while the virtual camera navigates around inside the nasal airway model. For adversarial image style transfer training, we firstly 3D printed a nasal airway phantom based on the digital nasal airway model. Endoscopic images are then captured inside the phantom airway with the Olympus laryngoscope. These endoscopic images provide a reference for the target domain, while the synthetic endoscopic images obtained from the virtual simulator provide a reference for the source domain. And this video shows how images are captured from the virtual simulator. And here is the qualitative illustration of the real-time prediction. Last but not least, after obtaining depth information, 3D annotation is performed. When a cursor is clicked at a pixel on an endoscopic frame, we get the depth value at that pixel, which is obtained from monocular depth estimation. Second, of course, we need the endoscope camera intrinsic parameters to map a point on the 2D image plane to the 3D physical world. However, the 3D position is now with respect to the camera. We want it to be static with respect to the physical world frame of reference. Thus, the camera pose with respect to the physical world frame reference is required to do this conversion. To track the camera pose, an NDI Aurora EM tracking platform is used. Also, standard hand-eye calibration is carried out to find the transform between the camera and the EM sensor. Therefore, the net effect is that when the cursor is clicked, a virtual annotation would be instantiated in the physical world frame of reference. So this is a video showing the process of instantiating 3D annotations by moving the laryngoscope inside the nasal cavity of the phantom. It is important for 3D annotations to be placed with fairly accurate depth and kept steady. So after achieving 3D annotation with predicted depth, we carried out both quantitative and qualitative evaluation on depth accuracy and stability. Regarding depth accuracy, we firstly registered the virtual 3D nasal airway model onto the 3D printed phantom, which was placed as static inside the EM tracking field. Ground truth depth maps were then captured based on the structure of the registered virtual model, while corresponding endoscopic RGB frames were also captured simultaneously. When evaluating the results, we adopted evaluation metrics from previous related studies, which are the normalized root mean square error and structural similarity. Results showed that our method achieved a very high accuracy, with a structural similarity of around 0.831. Though we observed that when the endoscope gets closer to the surface, lower accuracy it gets. This is in part likely due to light's intensity saturation, yielding an average depth biased towards a high proximity value. For stability, we evaluated consistency of depth predicted in consecutive frames. In this evaluation, we placed a virtual sphere at a known position and recorded its depth on the endoscopic frame when the endoscope undergoes this forward and backward motion. We call this reference depth. At the same time, the predicted depth at the same reference point was recorded. Average mean absolute error between reference and predicted depth of a total of 5 trials ranges from sub-millimeter to 2 millimeters. It shows a high accuracy but a low precision. It is a limitation to be noted as prediction made by supervised monocular depth estimation often flickers due to independent per frame processing. Moving forward to future work, we are interested in using self-supervised depth and pose estimation. In the process, preparation of ground truth depth is not required as the supervisory signal is obtained via image reconstruction. As it does not require an extra image style transfer network prior to depth estimation, computational complexity is also reduced. We are also preparing for a cadaveric dataset for training and testing in order to provide a proof of clinical feasibility. Here is a figure briefly illustrating the workflow of using a self-supervised depth and pose estimation method. While predicted depth can still be used for making 3D annotations, we can also take advantage of predicted pose to further stabilize camera pose measurement by the EM sensor. Because existing EM tracking is susceptible to interference from ferromagnetic materials or EM fields. And this is the end of this presentation. Thank you very much for your attention.